In today's video we are going to discuss about the types of antibiotics. Okay, so in the previous video we had like a brief introduction regarding antimicrobials, antibiotics. So today we are going to discuss the second or one of the most important questions that comes in your exams is types of antibiotics, right? So the types of antibiotics, you can classify antibiotic based on in multiple ways, right? But the most important classification is the classification that is based on their mechanism of action. Alright, so we'll be discussing antibiotics based on mechanism of action. So based on mechanism of action, you can divide antibiotics into 6 types, right? So 1, 2, 3, 4, 5 and 6. So in this video we are going to go briefly into each kind of antibiotic, and this is a really important video because if you understand this concept, like the brief introduction of each mechanism, then when you study individual antibiotics and the individual drugs, then it's going to be a lot easier, right? So the first kind of antibiotics is the one that works by inhibiting cell wall synthesis. Okay, so in order to understand this, we need to understand the structure of a bacteria, right? So this is your bacteria, right? And okay, hold on, I'll make it a little bit bigger. So this is your bacteria, and bacteria has a cell wall, and so human cells don't have cell wall, but bacteria have cell wall, right? I think you know this, right? So this is your cell wall. So the bacteria, apart from having a cell wall, it also has a cell membrane as well, right? So and then it has a it does not have a nucleus. Its DNA is outside. It's called a nucleolus or something, nucleolus or nucleoid. It has a nucleoid, right? And it has some ribosomes to make proteins for the bacteria, right? Because bacteria needs to make proteins, enzymes for it to survive. So drugs like penicillin, cephalosporin, carbapenems, monobactam and vancomycin. So in this group, just remember penicillin and cephalosporin. So drugs like penicillin and cephalosporin, how do they work is they simply go, so this is your penicillin or cephalosporin, they go and destroy the cell wall, right? So whenever this cell wall is destroyed, right, so what was the function of this cell wall? The bacterial cell wall was mainly present there to maintain the shape of the bacteria, right? So without the cell wall, what would happen is, if the cell wall is not present, inside the bacteria there is lots of proteins, fluids, ions, right? So it's osmotically active. So if there is no cell wall, then water from outside the bacteria will go inside the cell, and it will make the cell swell, and then it will make the cell burst open, right? But since the bacteria has this rigid cell wall, this cell wall is literally just holding the pressure, preventing the cell from swelling and being burst open, right? So if we give this drug penicillin or cephalosporin, and if it comes and it destroys the cell wall, or it destroys all the cell wall, then without any cell wall, there is no cell wall holding the bacterial cell together. So water from outside comes inside, right? And then the cell swells up, and then it bursts open, and the bacteria dies. So that is one mechanism of action of how penicillin and cephalosporin works, right? So that is one mechanism of action. So another mechanism of action is antibiotics can work by damaging the cell membrane, right? Now this is not as common, right? So this is not as common, but just for the sake of this video. So apart from the cell wall, we know that the bacteria also have this cell membrane, right? The one that I've highlighted in blue. So this inner membrane is the cell membrane, right? So similar story with cell wall. All cells need to have cell membrane, right? So if you destroy the cell membrane, then the stuff that is inside the bacteria that was supposed to be within the bacteria is going to leak outside, right? So if the internal cell structures leak outside, then the cell dies, right? So that is another way by which antibiotics can work. So these antibiotics destroy the bacteria by damaging the cell membrane. Examples are polymyxin and bacitracin, right? Another probably more important mechanism is inhibition of protein synthesis. See, this is one of the most important mechanisms by which antibiotics work, okay, and its examples are tetracycline, macrolides and clindamycin, right? So just remember tetracycline and macrolide, these are one of the most commonly used drugs, right? Macrolides include antibiotics like erythromycin, clarithromycin, azithromycin, all of these have the same mechanism of action. So how do tetracyclines and these protein synthesis inhibitors work is, as I told you, the bacteria also has this ribosomes, right? And the function of this ribosome is to produce proteins, and the bacteria requires to produce proteins continuously because bacterial proteins consist of enzymes, right? Main thing. So all enzymes are protein, right? So bacteria needs to continuously produce these enzymes, because without enzymes no chemical reaction can happen within the bacteria. So if there is no chemical reaction happening in the bacteria is going to die, right? So proteins are enzymes and the bacteria needs to produce protein. So whenever you give this drug such as tetracycline or macrolides, what do they do is, see, if you take this ribosome, right, and zoom it out, the ribosome has these two units, right? Units I forgot, like 30S and 50S unit, right? So what these drugs do is, the drugs such as tetracycline, right? Tetracycline goes and binds with the 30S ribosome, the 30S subunit of the ribosome of the bacteria, right? And it inhibits it. When the ribosome is inhibited, then no protein synthesis happens, right? And whenever there is no protein synthesis, there is no enzymes, and then whenever there is no enzymes, there is no chemical reaction and the cell dies, because it requires enzymes and proteins continuously. Similar to tetracycline, macrolides, right? Macrolides come and bind with the bigger 50S ribosomal subunit, right? So macrolides bind with 50S subunit of the ribosome, and they do the same thing. When it binds with the ribosome, it inhibits the ribosome, and whenever the ribosome is inhibited, there won't be any protein synthesis. Whenever there is no protein synthesis, the bacteria doesn't have enzyme and the bacteria dies, right? So one quick way to remember this is, the one that I use during my exams is, see this as macrolides, right? Macrolides, macro means big, right? Big. So that's why it inhibits the 50S ribosomes, whereas tetracyclines, it does not have macro, so it's small, right? So it inhibits the 30S ribosome. Just a quick mnemonic to remember, right? So we've discussed the important bit. Let me erase this a little bit, right? Now another way by which antibiotics can work is by misreading of proteins. Now this is also very, very important, right? Misreading of proteins and formation of aberrant protein, that is another classification of antibiotics. And the most important example is aminoglycoside, right? Such as streptomycin, gentamicin, kanamycin. There's a list of aminoglycosides. Again, these are one of the most important groups of antibiotics. So how do aminoglycoside work is, let me quickly erase this, right? So again, aminoglycosides work is, they work by binding with, they work by inhibiting, they don't inhibit protein synthesis. I mean, they kind of do, but the main mechanism of action of aminoglycosides is they cause formation of aberrant or wrong protein. Aberrant means wrong protein. So what do I mean by this? So again, this is your bacteria, right? And this is your nucleolus or nucleoid of the bacteria, right? The free floating DNA, and these are your ribosomes, right? So I told you these ribosomes continuously make proteins, right? So whenever you give this aminoglycoside comes inside the cell and it binds with, again, the 30S subunit, similar to tetracycline. See, tetracycline also binds with 30S, aminoglycosides also bind with 30S. So this aminoglycoside, it comes and binds with the 30S subunit of the ribosomes, and instead of inhibiting the protein synthesis like tetracycline, what it does is it actually confuses the ribosome, right? So the ribosome, right, so this is your 30S subunit, 50S, so it comes, binds with the 30S ribosome, and it confuses the ribosome, right? So whenever the ribosome is confused, instead of, so the function of ribosome is to make proteins, right? So whenever the ribosome is confused, instead of making the right protein, it instead produces the wrong protein, right? And this wrong protein is also called aberrant protein, right? Here's the spelling, aberrant protein, right? Aberrant, ABERRANT, right? Aberrant protein. And why is this important? Because whenever we confuse the ribosome, right, and the ribosome produces the wrong protein, this aberrant protein is toxic to the cell. So, and then because of the protein, the wrong protein production, this wrong protein is toxic to the bacteria and the bacteria dies, right? So this is how aminoglycosides work. So the aminoglycoside work by causing misreading of the proteins by the ribosome, confusing the microribosome and producing aberrant protein. That is another way how antibiotics can work, right? Good, right? So keeping all these things in mind, I came up with this whole Better Notes channel, YouTube channel. So apart from this YouTube channel which you are watching right now, I also have tuition. So I also give tuition. So if you're interested in tuitions, you can contact me. I also have a Telegram group where like almost 200 plus students from different medical colleges are already added in a couple of my Telegram groups, where they can ask me questions, or you can ask questions. I will try to help you guys in this Telegram group, or your friends might even help you. So it's like we have Telegram groups to discuss questions and like things you're struggling with in general. So make sure you join the Telegram group. And I have notes as well, the notes that I personally use for my own exam preparation, which a lot of my juniors absolutely love. Okay, thankfully they like it, right? So if you want these notes, and apart from the notes, I also have clinical videos. This is upcoming, right? So hopefully I'm also planning with my friends to make some clinical videos within the hospital itself, so that you guys can have a little bit more help on the clinical side of things as well. So if you are interested in any of these, the tuitions, the notes, the Telegram groups, all of this, the link to all of it can be found in the comment section, right? So the first comment in the comment section, you can find the links to all of it. So make sure you check out the comment section.